Good morning students, welcome to LEELA'S tutorial. Today we'll see the third generation molecular markers or the new generation molecular marker SNP, that is single nucleotide polymorphism. So these are the markers which help us to identify and differentiate between different traits or the variants, right? Let us see. The SNP is one base variation in a single DNA nucleotide that occurs at specific position in the genome. That means here if you see the complete genome, here I have shown the chromosome and here you can see the nucleotides, see, and I have shown only the gene coding sequence. So in this gene coding sequence, when I zoom this one, you can see this gene coding sequence where I have shown you the bases A, C, T. So the complementary bases are T, 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 G, A. That means A is complementary base to T, A to T, A, T, C, G and T, A. Right? So in this gene coding region, the common genotypes, that is A and T, it is common in all the genotypes. That means here in a single DNA nucleotide, a single base variation, right? So this single variation, if there is any variation in the single base, then you call it as SNP or single nucleotide polymorphism, right? So here this is a common genotype which results in a functional protein. That means here there is no any defect or no inefficient breed here. Right? It is having a functional protein, so it is a superior quality animal. Right? But here in this case you can see in its genome, again if I check the gene coding region, when I elaborate this one, see here instead of the same common gene, right? It is same gene which I am showing here. But here instead of A base, there is a variation. Instead of A, there is G, right? Because of which this complete nucleotide is getting changed only because of the single base. So here what is happening, when it is G, the complementary base, the T is not complementary to G. So what happens here, the C will become the complementary base to the G, right? As a result, the complete nucleotide has been changed. Right? So when this undergoes gene expression, it will result in some protein. But this protein will not produce the functional protein due to this SNP, the single nucleotide polymorphism. It will result in formation of altered protein. So with this altered protein, we will get a low quality breeding animals. Right? So here we want only the functional protein or beneficial animals in our farm or the livestock. So we have to check for this single nucleotide polymorphism which is occurring in certain animal variants. Right? So here main use, here when you check this, okay, when you check this, you know that some of the animals are having functional protein and some of them are having altered protein. So how you are going to know whether it is a functional protein or altered protein? So before you go for the product check, you can analyze it by using the samples directly. Right? So when you go for the agarose gel electrophoresis, you are going to take the samples, right? And you are going to make a well and load the sample, the A specific fragments, AT specific fragments and T specific fragments. So when you run the gel, again the bands will arrange themselves in a specific pattern with which you can know the A specific alleles and T specific alleles. How many alleles are present? The A specific and the T specific. With this you can know what is the SNP that is occurring in that particular animal. Right? So what is the use? Why you are using this SNP when you are having RFLP and RAPD? Because here even a minor changes or minor variations can be detected by using your SNP technique. see here, the SNP are most abundant and stable genetic variation that is occurring in the genomics. So it forms a very efficient tool for also selecting the farm breeds, right? That means the farm breeds which are having more beneficial characters. And then here this SNP also helps in prediction of individual response to certain drugs, chemicals and other substances, right? And then they also help in to check the disease developing breeds also, right? So when they produce the disease, when there is some variation in the some nucleotide, right, or in some bases, like that, even the risk of developing diseases, the animals which are having the risk of developing the diseases can also be known, right? Then it is susceptibility to environmental factors such as toxins and all. So if there is any toxin or food poisoning occurring, even that we can be known by using this simple technique. So this forms an very important technique, and the main application is SNP plays an very important application in animal breeding programs, right? So in order to know whether the animals are in completely superior quality or the inferior quality, that can be revealed. So nowadays they are using this third generation or the new generation molecular marker in order to know the best producing or best performing breeds. So here first application is they form a basic resource for selecting in the animal breeding program, right? Then it is used to study the quantitative traits of different variants, right? So you can know what is the quantity or quantity, and also the qualitative analysis can be done of those products, right? The products that are produced by these animals. So here SNP is very simple technique but most effective technique. Is that clear? If you have any doubts, put it in comments. Subscribe for the further videos.